So welcome to this Cooper Surgical Symposium, where we're looking at the subject of demystifying pre-implantation genetic testing for aneuploidy mosaicism. And we've got a great panel of key opinion leaders. We're going to be talking in particular about probably what's going to be looked upon in the future as a landmark paper, which is the data from the transfer of 1,000 embryos with known mosaicism. And we'll be talking to some of the authors of that paper and going through some of that in detail. So my name is Tony Gordon. I'm the Vice President of Global Genomics Business Development at Cooper Surgical. I will be the moderator of this. I have the pleasure of two founding fathers of our field of PGT, Mark Hughes, who is the Medical Director of Cooper Surgical. Also, Santiago Monet, who's the Chief Innovation Officer and co-founder of Overture Life. And Santi still works closely with us here at Cooper Surgical. And finally, Manuel Viotti, who I think is a rising star of this area of PGT. Manuel is a senior scientist at the Zuves Foundation for Reproductive Medicine, where he heads their sort of research into IVF efforts with special focus on molecular biology, cell biology, and the uterine environment. He earned his PhD in development biology from Weill Cornell Graduate School of Medical Sciences and the Sloan Kettering Institute. And since 2017, he's been key in the foundation of the Zuves Foundation. He also has published widely and is still an embryologist, still actively takes part in PGT-A and is a HCLD. So Santiago Monet and Mark Hughes have been in this field since the field started. So Mark Hughes really was instrumental in the molecular biology of the first PGT-M case. PGT-M is pre-implantation genetic testing for monogenic disease. So this is looking to transfer embryos without a known disease, and that was a cystic fibrosis case. It was the first successful case in 1992. And that has gone on to be a whole new field of medicine. Santiago Monet was the first person to successfully perform pre-implantation genetic testing for aneuploidy, PGT-A, and that's where you're taking an embryo biopsy and looking for the right numbers of chromosomes. And both of these gentlemen have been instrumental in the development of our field, and Santiago Monet is the last author of the paper that Manuel is going to be talking about a little bit later in this presentation. I'm just going to give you a little bit of background to what we're doing here at Cooper Surgical, because we are trying to also really innovate and bring new tests to this field. And we have a test called PGT-AI, AI standing for artificial intelligence. We have huge amounts of data here at Cooper Surgical. We do a lot of PGT-A cases, and we've been able to leverage that big data, terabytes of data, to use artificial intelligence and neural networks to improve the way we call this. So our calling is now purely mathematical, statistical. It is automated, so there's no human intervention. And we've innovated in things like establishing different truths. So where we used to use cell lines for our normalized baseline, we use actual embryos with known outcome. And our first iteration used 1,000 euploids that developed into live births or ongoing pregnancy. And we've done very large validations. This process can be kind of graphically shown here. So we would start off with an embryo biopsy and we'd amplify it like we have done for many years, about 1 millionfold. And then we put it onto these next generation sequences, not to sequence every base pair in the embryo, but to count the number of chromosomes in that biopsy. And we used to represent this as these charts which show up and down for gain or loss. And we used to have to subjectively interpret them from the scientists within the lab. Now that digital signal is not put into an image and then reported, but stays digital and goes into our algorithms and is statistically called and then automatically put into the report and then sent to the clinic, which removes subjectivity and human errors and makes the process consistent. We've also worked very hard to try and improve the accuracy of the testing. And one of the things that we've done, and one of the major challenges of using next generation sequencing, is mosaicism. And mosaicism is something that Manuel is going to talk a little bit about in his presentation. But essentially, instead of the embryo having all cells normal, euploid, or all cells or some cells aneuploid, you have a mixture of euploid and aneuploid, a mosaic, a mixture of these. And one of the first things we did when we developed this new algorithm, PGT-A, was to look at artificial mixtures of euploid and aneuploid cells. And this was a 270 sample experiment where we increased in purple the amount of aneuploidy in different mixtures. And you can see we can titrate down. The green line in the middle shows 20% gain or loss of chromosomes. So we can show the green line, which is euploid, from mixtures of aneuploid going up to the point where you would actually have all cells aneuploid. So this is really important because actually calling mosaics is much more difficult than calling full aneuploidies, and we need to be accurate. And we've been able to set our clinical resolution at 20%. And this is something that Manuel is going to talk about in his presentation. So just a brief summary of PGT-AI. It uses automated AI calling algorithms to improve accuracy, reduce subjectivity, and reduce errors. Another innovation that I haven't really had the chance to talk about, but is that we use two processes to call any change. We count the chromosomes using the next generation sequencing, and we also look at things called the single nucleotide polymorphisms, SNPs. And using that, we can detect triploidy and the origins of aneuploidies and some other things. And we've moved on to show that that has a high clinical impact and actually shown benefit in the clinic using this. And we're looking to continue to innovate and bring new features to this testing in the future. So without further ado, I would like to welcome Manuel to talk about his landmark paper. I think it's now, I think I can now say. And once we've gone through that paper, we will be discussing with Santi and Mark some of the really interesting points. And then once we've gone past that, we will be open to questions from the audience. So over to you, Manuel.
Hello, my name is Manuel Viotti, and I'm from the Zues Foundation. And first of all, thank you for the organizers for inviting me. Today, I will speak about our experience with the transfer of embryos that we classified as mosaic with PGT-A. And I'll be talking about this paper that was published earlier this year on our experience with 1,000 mosaic embryo transfers. And then I will go a little bit into data that is not published yet, that is on prenatal testing and neonate follow-up of babies that are born from mosaic embryo transfers. So nowadays, most of PGT-A is done at the blastocyst stage. This is an image of a day 5 blastocyst. And if all cells in the blastocyst are euploid, we get this kind of confirmation. All cells could be aneuploid, or sometimes some embryos are mosaic. And that means that there's a mix of chromosomal content. Sometimes the mix is all aneuploid cells, but different aneuploidies. And sometimes it's like it's represented here, a mix of euploid cells and aneuploid cells. And those are really interesting to us because as opposed to a few years ago where those were discarded or not chosen for transfer, nowadays the tides have changed a little bit and we are considering this in the clinic. And that's what I will talk about. So when we when we do PGT-A and we collect a biopsy, a multicellular biopsy, what we get in this case is a profile that is consistent with mosaicism, where some of the cells are euploid and some are aneuploid in the biopsy. Now, the question obviously is what to do with these embryos and how do we manage them? Like I said, up to 5 years ago or so, the consensus or the most of the stance towards those embryos was to discard them because there was evidence of aneuploidy. But this report from a group in Italy was the first one to show that that type of embryo can be transferred, and not only that, it can lead to what seemed like healthy pregnancies and healthy births. Here, the authors transferred 18 embryos into 18 women, and those embryos had the Mosaic profile, and 6 of them led to healthy births, apparently healthy births. So that started a trend in clinics across the world, and several have published their experience with the transfers of such embryos. Using the different studies use different PGT-A platforms. They use different definitions to define or to categorize mosaic embryos and different sample sizes. But overall, they all agreed in unison that mosaic embryos or embryos with that mosaic profile don't perform as well as euploid embryos. So the success rates in terms of implantation and achieving an ongoing pregnancy were lower in the mosaic. It was lower in the mosaic group. However, there were some discordances regarding the features or the characteristics of mosaicism detected with PGT-A and how they correlated with clinical outcomes. So what we decided to do is a couple of clinics and centers really spread out across the world decided to pool our data together. And when we reached 1,000 mosaic embryo transfers, we decided to analyze it and see if such trends were present or not, such trends of mosaic traits and characteristics with clinical outcomes. These are the participating centers and clinics. We all use the same PGT-A platform, and that's a next-gen sequencing-based platform, which in the meantime is pretty obvious is superior to ARRAY-CGH or other platforms, and is really the most indicated one to detect mosaicism correctly. We used the same definition, same thresholds to define a mosaic result, and that's the famous 20 to 80% as suggested initially by PGT-A and so on, and followed by many clinics around the world. There was genetic counseling with every mosaic embryo transfer. And the recommendation was to consider amniocentesis, prenatal testing, but especially amniocentesis. And we obtained IRB approval for this study. So here, initially, the results, just comparing the overall euploid control group, and there were over 5,500 euploid embryo transfers in this control group, compared to the 1,000 mosaic embryo transfers. And here we're considering all the mosaic embryos, regardless of the subcategory of mosaicism. And there's a difference in implantation rates between the two, statistically significantly so. And that difference is even more pronounced when we consider only mosaic embryos that involve whole chromosomes as the aneuploidy, so excluding those of segmental mosaic patterns. Using another clinical outcome, ongoing pregnancy or birth at the time of analysis, these trends are even more pronounced and more significant. When we look at spontaneous abortions of implanted embryos, and here we're really considering any spontaneous abortion, so any embryo that implanted and showed a gestational sac, but then was lost during the pregnancy, even, for example, at the time of fetal heartbeat monitoring, we're considering that a spontaneous abortion. So in the euploid group, the rate of loss is 8.6%, but in the Mosaic groups, it goes up to 25% if we consider only the mosaics that involve whole chromosomes. So obviously, when we compare two groups, the euploid group to the Mosaic group, there could be confounding factors between these two groups that could explain the differences we saw in clinical outcomes. So we looked at some of the usual suspects. So we looked at first day of biopsy, whether on average they were different between the euploid and the mosaics, and they were roughly similar. The maternal age was also the same between the mosaic embryos that were transferred and the euploid embryos that were transferred. However, the average number of blastocysts in the cycle was different between the two groups. So when a mosaic embryo was transferred, it tended to originate from a cycle that produced fewer embryos and especially fewer euploid embryos. And that's to be expected because usually the euploid embryo gets transferred first, or and in the absence of euploid embryos, then the mosaics were considered. The indication for PGT-A was roughly the same between the euploid and the mosaic embryo transfers. However, the morphology of the blastocyst was different between the two groups. So when we break down the morphology by the Gardner and Schoolcraft stage, ICM grade, and trophectoderm grade, we see, for example, that mosaics, there's a higher incidence of C grade mosaics compared to the euploids, and a higher incidence of C grade in the trophectoderm as well, not only the ICM, but the trophectoderm as well. So these differences in morphology could really be explaining the differences we saw in clinical outcomes. So we needed to normalize for these differences to ensure that it wasn't the morphology that was creating those differences in clinical outcomes. And to do that, we initially, as I said, there was a difference in morphology between the euploid and mosaic groups, but we created in a way, an artificial control group of the euploids, basically extracting data from our euploid embryo pool that perfectly matched the morphology of the mosaic embryos. So here, it's perfectly matched, and we can now do the comparisons in clinical outcomes. And now the euploid group is perfectly matched, and still we see these trends, meaning that the differences in clinical outcomes between the mosaic and the euploid group was not merely due to differences in morphology, and something else is going on, which we think is related to the chromosomal content of the embryos. Another thing is that since this was a retrospective analysis, obviously there's always the risk of bias in retrospective analyses. But it turns out that of the 1,000 embryos, the mosaic embryos that were transferred, 16.4%, so therefore 164 of them, those embryos, were transferred without the prior knowledge of mosaicism by the physician. So they were basically transferred as if they were euploids. And it turns out that that subgroup of the mosaics performs just like the overall mosaic group combined. And this gave us confidence that there wasn't bias related to our analysis, our retrospective analysis. This is the meat of the study, because now if we consider the subgroups of mosaicism, of mosaic embryos, because there's two features of mosaicism that can be learned from PGT-A. One is the level of mosaicism, which is, in other words, the perceived or the percent aneuploid cells that are present in the multicellular biopsy, which can be low or high, and the type of mosaicism, basically which aneuploid is involved in the mosaicism. Is it segmental, 1 or 2 chromosomes, or complex, where more than 2 chromosomes are involved? And so a mosaic embryo can have any combinations of these features. And when we plot the data by the different subtypes of mosaicism, so here on the left, we have implantation, and on the right, the ongoing pregnancy or birth. And on top, we have the euploid control group, and then all the different permutations or groupings that can be created by the different of mosaicism. And when we plot plotted this way, we see that the different subgroups perform very differently. For example, the high mosaics tend to cluster at the bottom and perform the worst compared to the low mosaics, but the segmentals perform a little differently. They're the best performing, if you will, mosaic subgroup. And low mosaic segmentals and high mosaic segmentals perform similarly, interestingly. However, it's important to note that even the best performing mosaic group, which is the low segmental group, performs significantly worse, or is significantly less successful than the euploid control group. And on the other end of this range is the high complex mosaics that perform the worst. However, they still occasionally lead to healthy pregnancies and births, and should not be discarded. So what this tells us is that the classical binary result system of PGT-A of euploid or normal and aneuploid abnormal should really not be used anymore. We should consider this mosaic subgroup or a third group of mosaicism, because if we don't do that, either these embryos are either then grouped with the aneuploid group and they're discarded, so we would be discarding perfectly viable embryos, or they would be grouped with the euploid group and bringing down the success rates of the overall euploid group, since on average they perform worse. So either way, it will be a disservice to our patients. Furthermore, the mosaic group should be divided into subgroups according to the features of mosaicism, and a priority for transfer emerges. Now, there's three simple questions. Does this complicate things? Absolutely yes. And is it annoying? Yes, it is. However, is it worth it? Absolutely yes, because the reasons we just discussed. We don't want to discard perfectly viable embryos, but we also want to not drag down the success rate of the normal group. However, in the clinic, we know that the reality is a little bit more complex, and we're not just dealing with the results of PGT-A. There's also morphology, and we're ranking and selecting embryos by morphology as well, even if we just considered the euploid embryos. So what we did is we developed this matrix it lists the different types of mosaic subgroups, and then for each embryo also the morphological features by stage, ICM grade, and trophectoderm grade. And basically for any mosaic embryo, one can, using this table, come up with a value that can be used to rank embryos. One being the best, and labeled here in green, and red being the worst. So to make this simple, we created this app that is freely available, and using drop-down menus, one can select the features of any mosaic embryo, and then compare these values for several embryos within a cohort, and come up with some indication, some guideline of which one is most likely to lead to successful clinical outcomes, based, of course, on our experience of the 1,000 mosaic embryos transfers. Now, of the embryos that implanted, of the 1,000 mosaic embryos that implanted, about 80% were ongoing after 20 weeks. And of those, almost all were born or were late ongoing. We were able to collect prenatal testing for 30% of them being amniocentesis. Of those, 4.1% had UPD analysis for the chromosome that had been detected as mosaic during PGT-A. Another 16.7% did CVS, and about 10% did NIPT. And what's interesting is that the vast majority was normal of these prenatal tests. 5 cases were abnormal. Those were all 5 were amniocentesis results, abnormal results, which what they were segmental imbalances. And those segmental regions were smaller even than the resolution used during PGT-A. So they were these abnormalities were unrelated to the mosaicism observed during PGT-A. And in fact, in none of the cases, in over 100 amniocentesis and about 200, more than 200 prenatal testing results, none of them reflected the mosaicism that was detected during PGT-A. And we think this has to do with these self-corrective mechanisms that have been proposed, and there's mounting evidence from several labs showing that those actually do work. So basically what it means is that even though a subset or a mix of cells, there's a mix of cells at the blastocyst stage of normal and abnormal, but then the normal ones outcompete the abnormal ones during development. They proliferate faster. The abnormal ones are undergoing selective apoptosis and cell suicide, which means by the time prenatal test gets done later in the pregnancy, the abnormality is gone and and diluted out, and only normal cells are left. In fact, when we looked at features of the newborns of mosaic embryo transfers and compared them with newborns from euploid embryo transfers, and these are matched by a series of characteristics like a maternal age, things like number of embryos in the cycle, and indication for PGT-A. So we try to match them as perfectly as we can. We see that, and this is still a mounting. We're still collecting more data for this, but at this point, of 162 babies that were born from mosaic embryo transfers, they have roughly the same birth length of gestation and the same weight at birth than their euploid counterparts. And furthermore, by routine inspection by the physician at birth, none of them had obvious gross abnormalities. So from this, we surmise that the detrimental effects of embryonic mosaicism detected with PGT-A really manifest themselves, is mostly manifested early in the pregnancy, either by decreased implantation rates or early increased rates of early spontaneous abortions. However, as the pregnancy progresses, by the time the babies are born from mosaic embryo transfers, there's nothing perceivable left that differentiates them from the euploid embryo transfers. And with that, I just want to thank all of you for your time, and of course the participating centers in this study. And thank you very much.